Hello everyone, I'm Shravya Jasti. Let's today discuss about gene cloning and the steps of gene cloning. Gene cloning is a common practice in molecular biology. It is used to create copies of particular gene for downstreaming applications such as sequencing, mutagenesis, genotyping, or heterologous expression of a protein. The traditional technique for gene cloning involves the transfer of a DNA fragment of interest from one organism to self-replicating genetic elements such as bacterial plasmid. What is gene cloning? The production of exact copies of a particular gene or a DNA sequence using genetical engineering techniques is called gene cloning. The term gene cloning or DNA cloning or molecular cloning and recombinant DNA technology all refers to same technique. When DNA is extracted from an organism, all its genes are obtained. In gene cloning, a particular gene is copied, forming clones. Cloning is one method used for isolation and amplification of gene of interest. What is a vector? Well, a vector as related to molecular biology is a DNA molecule, often plasmid or a virus, that is used as a vehicle to carry a particular DNA segment into the host cell. As part of cloning or recombinant DNA technique, vectors can replicate autonomously and typically include features to facilitate the manipulation of DNA as well as genetic marker for the selective recognition. The most common vectors are DNA plasmids, virus, and artificial chromosomes. Here are the following steps involved in the gene cloning. Firstly, isolation of donor DNA fragment or a gene. Secondly, selection of suitable vector. Thirdly, incorporation of donor DNA fragment into the vector. Fourth step, transformation of recombinant vector into a suitable host cell. Finally, isolation of recombinant host cell. Now let us discuss each point clearly. First step, isolation of DNA fragment or a gene. The target DNA or a gene to be cloned must be first isolated. The desired gene may be isolated by using restriction endonuclease enzyme, which cut DNA at specific recognition nucleotide sequence known as recognition sites towards the inner region, producing blunt or a sticky ends. Sometimes reverse transcriptase enzyme may also be used, which synthesizes complementary DNA strand of the desired gene using its mRNA. Coming to the second step, selection of suitable cloning vector. The vector is a carrier molecule which can carry the gene of interest into a host, replicate there along with the gene of interest, making it multiple copies. The cloning vectors are limited to the size of interest that can carry. Depending on the size of application of interest, the suitable vector is selected. The different types of vectors available for cloning are plasmids, bacteriophages, bacterial artificial chromosomes, yeast artificial chromosomes, and mammalian artificial chromosomes. However, the most commonly used cloning vectors includes plasmids and bacteriophages beside all the others available vectors. The third step is incorporation of donor DNA fragment with the plasmid vector. The plasmid vector is cut open by the same restriction enzyme used for isolation of donor DNA fragment. The mixture of donor DNA fragment and the plasmid vector are mixed together. In the presence of DNA ligase, base pairing of donor DNA and fragment and plasmid vector occurs, forming recombinant vector in the mixture. And the fourth step is transformation of recombinant vector into the suitable host. The recombinant vector is transformed into the suitable host cell, mostly a bacterial cell. This is done either for one or both following reasons. To replicate the recombinant DNA molecule in order to get multiple copies of the gene of interest, or to allow the expression of gene of interest such that it produces needed protein product. Some bacteria are naturally transformable. They take up recombinant vector automatically. For example, Bacillus, Haemophilus, Helicobacter pylori, which are natural competent. Some other bacteria, on other hand, require incorporation by artificial methods such as calcium ion treatment or electroporation, etc. And the final step is isolation of recombinant cell. The transformation process generates a mixed population of transformed and non-transformed host cells. The selection process involves filtering the transformed host cells only. For isolation of recombinant cell from non-recombinant cell, marker gene of plasmid vector is employed. For example, PBR322 plasmid vector contains different marker gene, ampicillin resistant gene and tetracycline resistant gene. When PST1 restriction enzyme is used, it knocks out ampicillin resistant gene from the plasmid so that the recombinant cell becomes sensitive to the ampicillin. Here comes the diagrammatic representation of the steps of gene cloning process. Firstly, a gene of interest is isolated from a cell, and on the other hand, a plasmid of interest is isolated. Now, combine the target fragment and the plasmid DNA, and then add the DNA ligase which closes the circle with covalent bonds, and put them into a bacterium by transformation and allow the bacteria to reproduce. The next step involves isolation of a multiple gene of interest attached with the vector or of the protein encoded by it. This is followed by purification of isolated gene copy or the protein. And here that's it, the process of gene cloning. Thank you.